Okay, good afternoon, everyone. My name is Vivek Goel, and I'm the Network Scientific Advisor for CanCOVID and a Professor of Public Health at the University of Toronto. And it's my pleasure to welcome everyone for this special workshop that we're holding with the task forces that the federal government has established in relation to COVID-19. And I really appreciate that the members of the different task forces, co-chairs, or in one case, the executive director, have agreed to take part today. The plan is to have about a 15 minutes maximum presentation from each of the task forces, and that should give us about a half an hour of time for discussion. I'm not going to do long, detailed introductions for our very illustrious speakers. We did include in the invite the link to the task force pages where you can look up the membership as well as their bios. And so I'll just introduce who will be presenting, and then we will start to go through the presentations. Feel free to post questions in the chat as we go along, but we will hold the discussion until after everyone has had a chance to speak. And I would also just remind everyone to mute so that we can ensure that we're able to hear the speakers. So the order we'll go in is with the COVID-19 Vaccine Task Force, which we're very pleased to have the co-chairs, Joanne Langley and Mark Luvinen, and Joanne will start off the presentation. Then we'll proceed with the COVID-19 Immunity Task Force with Tim Evans, the Executive Director, and then to the Testing and Screening Expert Advisory Panel with Irfan Dalla, who is the co-chair, and then conclude with the Therapeutics Task Force with Nancy Harrison and Cedric Bisson, the co-chairs. So with that, I'll ask Rosa to put up the first set of slides and hand it over to Joanne.
Well, good afternoon, everyone, and thanks so much for inviting Mark and I to present today. I'm here in Nova Scotia in Halifax, and I work at Dalhousie University in the IWK Health Centre. I'm a pediatric infectious disease physician and a vaccine researcher. And we're both delighted to be here to represent the task force. We'll tell you a little bit about that. And I just want to say, give Mark a chance to introduce himself because he'll take over halfway through the presentation.
Hello, everyone. I'm Mark Lavonin. I'm one of the co-chairs of the Vaccine Task Force, along with Joanne. I also chair the Joint Biomanufacturing Subcommittee, which I'll talk about in a minute. And I'm the former president of Sanofi Pasteur Limited and a senior fellow at the Della Lana School of Public Health. Delighted to be with you this afternoon.
Thanks,
Back
Mark.
to you, Joanne.
Great. Next slide. So this is this wonderful crew of Canadians who've volunteered when they were asked to do this work, and both Mark and I are just so grateful to be able to do this work with these people who represent diverse areas of expertise in immunology, vaccinology, biomanufacturing, company development, so many areas that are needed to understand how vaccines come from idea to injection or nasal administration or whatever the route is. And I think I can say with absolute confidence that every member of the task force is truly honored, and it's really a career highlight to be able to bring any skills we have to serve Canadians in this way. Next slide. So the work of the task force is strongly supported by a secretariat, and it is housed at the National Research Council of Canada, NRC. And the Secretariat brings together expertise from across the government, particularly in vaccine science, health policy, and project management. So the areas of expertise that are available that were brought together, and there's like over 30 people, allow them to apply areas of immunology, epidemiology, human health therapeutics, for example, our regulator, public health, knowledge of current biomanufacturing in Canada, what has happened so far in public health in the COVID-19 response, and various officials who know how to get work done, project management. Next slide. As you know, the mandate of the task force is to provide advice to the government on its COVID-19 vaccine strategy. That is, simply stated, to support the government to secure safe and effective COVID-19 vaccines for Canadians as soon as possible. So we were up and running by mid-June, and we focused our efforts on three critical short-term issues to meet this overarching priority. First, advising on priority Canadian vaccine projects, so that's domestic projects focused on COVID-19 vaccines. Second, identifying and meeting promising and bringing to attention promising international vaccine candidates, and then recommending concrete strategies to strengthen biomanufacturing here in Canada. And we've worked on these three streams in parallel. And the output of our work is that we provide recommendations to the Ministers of Health and Innovation, Science and Industry. And we also work fairly closely with the Procurement Ministry. So next slide. I'll just mention a bit about our domestic approach to vaccines and then turn the floor over to Mark, who will talk about the international approach to vaccines, as well as biomanufacturing for both therapeutics and vaccines. As I mentioned, the task force primary objective has always been to secure these safe and effective vaccines for Canadians with this time element as soon as possible. Whether the vaccines come from within Canada or abroad is really a secondary consideration, since getting them soon to mitigate this COVID threat is the chief priority. We acknowledge that Canada's vaccine and biomanufacturing capacity is limited. And so it was clear that the top prospects for a vaccine to get here for late 2020, early 2021, looking back at it in May, June of last year, would most likely be international candidates. Having said that, Canada does have a strong R&D effort in the COVID response. And so we worked with Innovation, Science and Economic Development, the ministry, I said, to review 24 domestic applications that were submitted to a request for proposals to develop a COVID-19 vaccine. We developed a framework to look at these proposals systematically and met with the proponents in many cases where this was necessary. And we focused on criteria such as the scientific merit, technical merit, project management merit, including the feasibility of getting a vaccine to market and the ability to scale up for millions of people, as well as to contribute to advancing Canada's vaccine landscape for the future. Ultimately, three proposals, Medicago, VBI, and PNI, Precision NI, were thought to show promise for a near-term and feasible response to COVID-19. And other vaccines candidates showed potential in terms of their science, but were at an early enough stage of development that it was clear they'd need more support to be able to develop a vaccine in a timely manner. So we recommended that 6 be transferred to the National Research Council Industrial Research Assistance Program, which is a longstanding program, to get this support to advance their development as quickly as possible. And those are shown on the bottom right there of your slide. While our domestic vaccine development efforts are not progressing as quickly as we might say they are in some other countries, Domestic candidates do remain an important aspect of our overall pandemic response and growing our vaccine research and development capacity in Canada. And to further elaborate on that, I'll turn the floor over to Mark, who'll talk about the international vaccine approach we took and biomanufacturing.
Great, thank you, Joanne. So if I could have the next slide, please. So as Joanne said, our mandate was to provide advice to the government of Canada on securing safe and efficacious vaccines as soon as possible. And it quickly came to our attention that as soon as possible meant our initial candidates would be from the international companies. And so we set out in the summer to review the most promising candidates and to assess those and to meet with the companies. Now, I think it's important to point out that back then, and the world has changed dramatically, what we know now versus what we knew in July and August. At that time, there was incomplete information. We adopted an approach of making decisions based on what we called battlefield readiness. So we were able to act and make it, give advice based on incomplete information, the best information that we had. And we met with the companies, we invited them in, we had a dialogue, we looked at a number of criteria, and you can see the framework on this slide for the things we looked at around scientific and technical merit, timely access for Canadians. And one of the issues was there a feasibility to produce in Canada, but that was not an immediate term aspect right away. A track record of these companies was important. We also looked at the supply chain. Very important to think about the supply chain. And even back then, there were discussions, you'll recall, about vaccine nationalism, particular concerns about our neighbor to the south. So we looked at supply chains, we looked at geographic location of supply chains, and we looked at the robustness of them. And one of my favorite parts was when we met with these companies, the capabilities of the task force and the members in terms of having a scientific discussion with them, a technical discussion, evaluating them. I think it went both ways. I think we learned a lot from the companies and were able to provide advice. I also think it served them well in terms of the discussions that we had. And I think they left those meetings feeling good. And that also put us in a favorable position to negotiate. And again, if you think about at that time, we were looking to provide advice to buy vaccines that had not yet been licensed. We weren't sure which would be licensed. And there was very much a risk assessment involved. So as a result of that, we adopted a portfolio approach, and I'm sure you've seen the communications around that. We recommended 6 international candidates across 3 platforms, being the mRNA vaccine platform, the viral vector platform, and the protein subunit vaccines. And we also were involved with COVAX and providing advice, and we do provide advice to the government related to COVAX and the international vaccines and those vaccines for vulnerable populations. So we recommended the six vaccines across the 3 platforms. Very pleased to say that to date, the 2 RNA vaccine platform vaccines have been licensed, Pfizer and Moderna, and those are underway. 2 of them that are under review include AstraZeneca and Johnson & Johnson, which you've heard a lot about, which are also in our portfolio. And then the protein subunit vaccines being Novavax and Sanofi GSK, which have suffered a few issues along the way, but could be there further out in time. And as I did mention, we're very much involved with COVAX. We also met with other international task forces, the UK, New Zealand, a group in Germany. We've had a number of ongoing discussions, and we started to get some confidence early on that our advice was resonating and was consistent, and we've continued to learn from one another and support one another. And if I could have the next slide, please. So our other major aspect, sorry, go back one, was biomanufacturing strategies. And as was mentioned, there was a joint biomanufacturing subcommittee comprised of members of the vaccine task force and the therapeutics task force. And so you'll hear from Cedric and Nancy later on, who are members of this group. I was asked to chair the group, so I'm chairing this one. And we really were looking at biomanufacturing and the issues to address, as Joanne noted, in both therapeutics and vaccines. We all have our knowledge of the history of vaccine and therapeutic manufacturing in Canada, where the situation is. Suffice it to say, there were no short-term candidates available, as we mentioned. And so we ended up providing advice on short-term aspects that had to be dealt with, which were things like procurement of syringes and swabs and vials. And we knew there'd be a worldwide competition for these kind of components. So to provide advice on that, we have provided advice on what I will call intermediate steps or intermediate capacity. So we've identified those firms in Canada that are doing some bulk manufacturing, drug substance, fill and finish, and they submitted SIF proposals. We've reviewed those and provided advice to the government of Canada on funding of those, and most notably the expansion of the National Research Council Biomanufacturing Facility in Montreal was advice that we gave and that has been acted upon to strengthen those components in the intermediate term. And we also have looked at the longer term, and we have provided advice to the government of Canada on a longer term biomanufacturing strategy for pandemic preparedness, so that in the future, not if, but when the next pandemic comes, that we could be ready for that and ready to have a made in Canada solution. So there's ongoing advice that's been provided in that area as well. And if I could have the next slide, please. So where do we stand now as we look ahead and plan ahead? So we have an ongoing monitoring of vaccine efficacy and safety. We continue to monitor the clinical trial results of the vaccine candidates that we've chosen. We continue to meet with the companies when it makes sense to do so, to evaluate their results and the efforts they have underway. We can refine our advice on an ongoing basis as we need to and should, and we will do that. We continue to engage on the international front. Canada has provided money through COVAX for both procurement of vaccines for domestic use and to fund the use of vaccines in international markets, international countries. So we continue to refine, we'll review those vaccine candidates as they come forward and provide our advice on what ones we think Canada should procure and should support. We continue to work in the domestic area, and we are able, as I said, to act on the basis of battlefield readiness and incomplete information. So we do work with the Public Health Agency of Canada and Health Canada and the other task forces and other groups. Joanne and I are involved with a number of other groups dealing with different aspects of COVID-19, so we continue to be available to do that. And we will continue to provide advice, refine our strategy on biomanufacturing, and including being a sounding board for the government as they go ahead and proceed with the advice and the implementation of the recommendations that we've made. So I'll stop there. Thank you for your attention, and Joanne and I look forward to any questions that you may have.
Thank you, Joanne and Mark, and thank you for sticking to your time. So we'll move to Tim Evans from the Immunity Task Force.
Great. Well, thank you very much, Vivek, and I hope people can see the screen.
Yep.
Very good. All right. Well, as everybody knows, we're well into the second wave, and the only reason I put this up is I don't think anyone in the world would have ever predicted this, and so we're learning as we go, and I'm sure we've got a lot more to learn moving forward. The Immunity Task Force was set up in late April of 2020 with really two fundamental aims. Is to understand the extent of SARS-CoV-2 infection across Canada, as well as the nature of immunity arising from infection. And our mandate has been to catalyze support and harmonize the implementation of studies that would provide reliable estimates of SARS-CoV-2 immunity, infection-acquired immunity, as well as look at the rates in specific subpopulations. This is the composition of the what we call the CITF leadership group. Unfortunately, we have too many members to have lovely pictures like the Vaccine Task Force. But this group is, I can assure you, as volunteers, just as dedicated and just as good looking. We have made scientific priorities in 3 primary areas in what we call phase 1. And the first was around through our field studies working party around seroprevalence studies to understand the levels and trends in immunity across the entire life course, age groups from youth or infants to elderly, in specific infected populations, follow those populations out over time and understand the evolution of immunity, indigenous populations, and then other high-risk groups defined occupationally, geographically, or otherwise. So those were what we call field studies, and those were underpinned by 2 types of other work that we promoted, which is more fundamental research on the nature of immunity and the immune correlates of disease, as well as focus on testing and the need to try and get accurate tests and get them to the places they were needed in order to do the testing. And you may or may not remember, but in April 2020, there wasn't one approved assay by Health Canada, and so there's been a lot of very collaborative work with the National Microbiology Laboratory. The phase 1 then covered these what we call convenience samples. We went to the places where we thought we could get blood easily, the blood banks, a lot of the antenatal surveillance, and leftover lab samples that are a part of the public health surveillance system across the country. We looked at trying to bring in draw on some of the population cohorts that exist, especially focusing on high risk or elderly through CLSA and other well-developed groups. But you can see here, we looked at priority populations, translational immune science, testing technologies, issued policy guidelines on testing for the appropriate use of serology tests. And that's been about over 50 investments to date and about $80 million in commitments. This is a list of the studies that have been supported. There are 55 in total, and some of them are listed institutionally here, but others came in batches. Most specifically, we co-funded with CIHR in their second phase of COVID financing in May. 22 studies, and then we also issued a call for hotspot proposals in specific geographies amongst high-risk occupations and in long-term care facilities, and financed 18 studies through that. Just some data. This was seroprevalence in May, June, based on Canadian Blood Services and Hema-Quebec data, and as you can see at that time, very low seroprevalence, less than 1% nationally. Quebec, the epicenter at the time, just over 2%. But the big message here, while Canadians have done a great job in mitigating spread in the first wave, however, we're dry tinder for the second wave, and we're nowhere close to herd immunity. We also launched an international effort called SeroTracker to understand all of the seroprevalence studies that were being done globally. And this dashboard has been tracking and mapping and assessing in a disciplined way studies so that they can be compared across geographies. That analysis has given actually very interesting understanding of the global epidemiology based on seroprevalence. And what we find is that overall, at least at the end of the first wave, these have not been fully updated, that seroprevalence was low when you did national studies. And as you did studies that were more focalized, you had higher seroprevalence rates in general. We have just had second wave results from our Canadian Blood Services study, and you can see that from May to November, seroprevalence has doubled nationally, but it still remains remarkably low at 1.51%. And again, indicates the necessity and imperative of accelerated vaccination. However, in the table, you do see a little bit of variation, more easily perhaps on this slide, which is the Prairies, Saskatchewan and Manitoba showed very significant increases in their seroprevalence over this time period, particularly between October and November. And very interestingly as well, Ontario's seroprevalence doesn't seem to have moved significantly. This is the temporal trend in the Prairies, and you can see that it was really very recent in the month of November, where the seroprevalence seemed to spike, at least move up very significantly to just under 10%. And we also have a view on cities, which is also interesting insofar as the cities in the West here all showed significant increases in seroprevalence between May and November, doubling, tripling, quadrupling. But in the East, Ottawa and Toronto, for example, what appears to be a flat level of seroprevalence. So again, these data provide interesting variations and ones that require a lot of further development. Just to say that we work through research networks. We've got a very vibrant pediatric children's network. We've got a network of folks that have cohorts of patients with COVID, and a whole set of studies that are focusing in on correctional facilities. Our hotspot studies have been really interesting because they are really trying to target in on the areas of greatest risk. And those are the studies, areas that we've been financing. Just one example of this, which gives you a sense of the science, borrowed this from one of the studies we're supporting at McMaster University. But it's really a very interesting effort to bring together dimensions of facility risk and the structure, the size, the staffing, financing of facilities, and correlate that with immune risk. And so this is just a taste of the design of the studies that are underway. In October and November, we decided we need to develop a phase 2 set of priorities, and that related to the fact that seroprevalence and infection-acquired immunity was not going to get us to herd immunity quickly, and there was real indication that vaccines were on the horizon. And so we started looking at how we could articulate important questions that would be worthy of focus of the CITF. And these were some of the areas that we felt were important at that time, and ones that the leadership group in November decided would be worthwhile focusing on in a phase 2 of the CITF. One of those areas was on vaccine surveillance, and today there's a presentation going on with the chief medical officers, with the 2 co-chairs of the Vaccine Surveillance Reference Group. It was created to try and help develop a comprehensive approach to vaccine effectiveness and safety in Canada, and in recognition that the phase 3 trials didn't cover everything. And it's a collaboration across institutions that are actively engaged, including the Canadian Immunization Research Network and the National Advisory Committee on Immunizations. The mandate is to advance support for vaccine surveillance plans across Canada in coordination with federal, provincial, territorial institutions, and with attention to all priority groups, including Indigenous persons, to monitor safety and effectiveness of Canadian vaccines. So there's a large set of priority population groups that have been identified, and these have been tagged and are being understood in the context of research priorities that NACI has issued and has updated recently on the sorts of questions that need to be asked from both a safety and effectiveness perspective. These are the members, and again, I apologize, no pictures, but we have Scott Halperin and Carolyn Quach, who are co-chairing this, and a number of those people. Importantly, we have a working group on vaccine effectiveness and a working group on safety. Each of those is co-chaired by Jeff Kwong and Karina Top, respectively. So I'll stop there, and I look forward to questions, and thanks very much, Vivek and CanCOVID, for the opportunity to present.
Great, thank you, Tim, and we're already getting some interesting comments going in the chat. So we'll go next to Irfan Dalla on the testing and screening panel, and also note that Irfan has posted their most recent report at the top of the chat for those that are interested. Irfan.
Thanks, Vivek. Thanks, CanCOVID, for the opportunity to be here today. Nice to see some familiar faces on the screen. I don't have any slides. The testing and screening expert advisory panel was established by the Minister of Health in November. Sue Paish from Vancouver and I have the pleasure of co-chairing the panel. Like I think the other task forces and panels, there's an incredible breadth and depth of expertise from coast to coast. We have an exceptionally effective and hardworking team at Health Canada, and we're also the beneficiaries of some very hardworking and intelligent students who've been supporting us in the background. So just wanted to acknowledge both of those groups, and of course the panel, including its ex officio members. I think we started from the perspective that we were a little bit behind. We were established in November, and obviously the pandemic has been raging since March. So we met quite a lot in November and December. The website says we met 10 times, and there's short summaries of the meetings on the website. Feels like we met even more than that, but I think there are probably some side briefings and technical briefings that aren't on the website itself. We submitted our first report in late December, and it was released publicly in early January, and that's the report I uploaded, and it's on the web as well. In our first report, we made recommendations in 4 areas. The first is to optimize diagnostic capacity with laboratory-based PCR testing. In some jurisdictions, the turnaround times are still quite long, and there are capacity constraints. And so we recommended that where that's the case, we encouraged where that's the case, the creation of higher priority and lower priority streams. So for example, higher priority streams might be used for tests that are coming back from long-term care homes where there's an outbreak, and a lower priority stream might be used for screening if laboratory-based PCR is used for screening, meaning testing in people without symptoms and who are not close contacts of someone with COVID-19. We also suggested a greater use of task shifting to increase capacity, both for specimen collection and also for test processing in the laboratories. The second area where we made recommendations relates to rapid tests. Both rapid molecular tests that are used at the point of care and rapid antigen tests. We recommended that the rapid tests be used to screen for infection, and that the rapid antigen tests be confirmed with PCR as appropriate. And there's a little bit of an algorithm in the report that suggests how we think this should be done. And I guess the main principle here is that in many settings, including essential workplaces and in congregate living settings, a rapid test is better than no test at all, even though the rapid tests, particularly the rapid antigen tests, have less sensitivity or reduced sensitivity compared to the PCR tests. The third area where we made recommendations relates to equity, both for testing and screening. It's saying the obvious at this point to say that the pandemic has revealed a lot of inequity in health and healthcare in Canada, and the pandemic has amplified that. And so we suggested, and that's been the case even when it comes to testing and screening, where access to testing has been much worse in some communities than in other communities, despite higher case counts and greater morbidity and mortality in these communities. So we suggested that both lab-based PCR and rapid tests should be leveraged to fill in testing gaps in particular geographic locations and in communities, and that it's important to work in a context-specific way to decrease barriers to testing in communities, working with community leaders, communicating in different languages and across various platforms. And we also threw in a little bit of a read between the lines recommendation about sick pay, paid leave for people who need to get a test or people who need to isolate or quarantine while they're waiting for their test result or if they're positive. And then the last area where we made recommendations relates to communications. It's been fascinating to me to see how testing volumes have come down so much in the recent weeks as vaccination has sort of dominated the communications narrative. Based on test positivity and current case counts, we probably need to be testing, or not probably, we do need to be testing much more than we are testing now if we want to control the pandemic. And part of that is about communication. I think we all still hear from friends, family, patients, whoever, that they don't know exactly when they should get a test, when they don't need to have a test. I think there is still, in large chunks of the population, this notion that the testing is for me. I get a test for me, and it really hasn't fully sunk through that for most people, the reason you're getting a test is actually for the community so that you can decrease transmission to other people. That's basically it in a nutshell. I think it might be, I didn't prepare slides and I don't have 15 minutes of material, but hopefully we can take some questions in the Q&A, and happy to try to answer questions in the chat as well. Thanks again, Vivek.
Thanks, Irfan, and I'm sure there'll be some discussion, and it's great to have extra time for discussion as well. So we'll go to our last presentation from the Therapeutics Task Force from Nancy and Cedric. I think, Nancy, you're going to start this one off.
Yes, I am. Thank you very much, Vivek. So my name is Nancy Harrison, and I am based in Vancouver. My background is as a repeat entrepreneur and a venture capitalist in the life sciences community. Maybe I'll just quickly ask Cedric to introduce himself, and then we'll pass it back and forth.
Thank you so much, Nancy. So I am Cedric Bisson. I'm also a venture capitalist and a medical doctor. I've been in the business of drug development for the past 30 years or 25 years. And I'm delighted to have a chance to speak with you today. Over to you, Nancy.
Yes, thanks, Cedric. So I speak on behalf of Cedric and myself and all the members of the CTTF. Maybe we can bring up the slides while I'm speaking. And we're very honored to be providing advice to the Canadian federal government. And I'll go through a little bit of how we approach this, like Mark and Joanne did, and our mandate. But suffice to say that the mandate originally and remains today, that we're hoping and advising on the evolving landscape of available molecules to treat COVID-19. And obviously this is an evolving situation, and our goal is to find the right therapies to gain access for Canadians to therapeutics as they relate to COVID-19. So the next slide just outlines our mandate. And our mandate is to provide this advice to the government along the same lines that Joanne and Mark covered. We're providing that on therapeutics. As one might anticipate, this is an evolving landscape. As clinical trial data becomes known, we were constituted in July. And so at that time, there was limited evidence and information around clinical trials for COVID. But as time has gone on, that's obviously becoming more relevant. And we track probably about 150 different therapeutics that are in clinical development. Our first priority has really been looking at therapeutic projects seeking support through Canadian government funding for activities in Canada. The second priority was really looking to gain access for non-Canadian therapeutics of interest to partner and procure for Canada. And the third priority has really been providing advice on the systemic issues that we have on delivering COVID vaccine therapeutics or therapeutics to Canadians. And that includes some of the items that Mark mentioned on domestic manufacturing, organization of clinical trials, stage of development, and Cedric will speak a little bit more about that. Our approach has really been to provide strategic advice to the Canadian government. And we really have taken, as I described, a portfolio approach, thinking about the various aspects of the disease as someone has COVID, the various subpopulations, be they gender, age, race, pregnant women, etc. The mechanism of action of both the disease and the possible therapeutics, and the stages of the patient's journey, from a prophylactic approach to somebody who is now on ventilators, and what would be the approach for that individual. So our original mandate was for 6 months. It was from June to November. It's been extended to the end of February at this point. So next slide, please. Our structure is quite similar to what, again, Mark and Joanne mentioned. There are 7 core members, including myself and Cedric. The other group of 5 individuals really represents people from across Canada, various scientific groups, as well as people who are company operators and used to triaging and looking at various aspects of information within a clinical trial data set. We're supported heavily by our science advisors, and I must say it's been an absolute honor and pleasure to be able to co-chair this group of scientists. These are acting clinicians focused in all the aspects of this disease, and great scientists, inflammation, infectious disease, chemistry expertise, a really rounded group of individuals that is coast to coast. And so it's really been a pleasure to be able to interact with them. And while we call them core members and science advisors, there really is no differentiation between these two groups of people in our mind. We're also supported by a great secretariat. We report and are housed underneath the Innovation, Science and Economic Development arm of the federal government. And so our secretariat resides in that group. So there's quite a good group of people who help support us. As one might imagine, we have a lot of tracking that we're doing in terms of clinical trials and interaction with companies, and this group at the Secretariat really helps support that. We have 4 ex officio members, so the Deputy Minister of ISED, the Deputy Minister of Health, the President of the Public Health Agency of Canada, and the Chief Science Advisor. And we regularly have, as all of our groups have, lots of other members from those organizations participating, listening in, helping at our meetings, which we're really thankful for. As the Vaccine Task Force mentioned, we've also met internationally with many other groups that also are focused on therapeutics for COVID, including the UK, France, Germany, as well as other countries. When we look at sort of where we're at in our mandate, we've had over 31 meetings since mid-July. We've offered 11 advice letters to the ministers. We've assessed over 40 Strategic Innovation Fund proposals from Canadian companies, and we've assessed at least 40 international therapeutic candidates. That's after we actually triaged everything that was in a clinical trial for COVID. We wanted to really assess 40 projects individually. We took an approach of really looking at thematic areas. So for example, we would look at all the antibodies focused on spike, or all the JAK inhibitors, the protease inhibitors, plasma, etc. And we reviewed that scientifically for its merits and its understanding around the disease and the mechanism of those various products and targets. We also have offered advice on strategic and high-level considerations, as Mark and Joanne talked about, how to best avoid some of the issues that we saw, how best to organize ourselves in case there is another pandemic in the future. What are our learnings broadly? Mark mentioned we also participate in the Joint Biomanufacturing Subcommittee. So we've not only thought about manufacturing around vaccines and large molecules, but also small molecules and how to approach that. As part of it, we've also referred several projects to other organizations like IRAP, as well as the Strategic Investment Fund and CIHR, etc. We've also advised on various other shortages of drugs that might relate to use as somebody has COVID. So for example, the drugs that are often given to people once they're on ventilators, or the devices one needs to actually administer therapeutics, and making sure that we don't have a shortfall in those. We've also participated in many of the COVID initiatives, such as Vivek organizes, or I mentioned CIHR. So we participate and come with our lens based on the therapeutics. Again, our goal is really to find the right therapeutics and access for everybody in Canada for these various people. I'll turn it over to Cedric to offer any other observations as we've offered advice to the group and to the government.
Thank you very much, Nancy. A couple of other observations for the group. Obviously, when we came into this situation, it was a well-known fact across Canada that the very novel nature of the SARS-CoV-2 virus was created severe limitations in terms of access to biological samples or access to animal models. So that was initially, and currently, still today, is one of the things that we have been working on, or actually providing advice on, to make sure that or to increase the propensity of Canadian companies to be able to work on preclinical models or in vitro models of the actual disease. As we looked at various proposals, it also became quite apparent that, with a few exceptions that are very well financed, most of our companies in Canada tend to be underfinanced. And so it's not really a level playing field versus international companies. And so far, there is a ton of great innovation across Canada. But if you look internationally, most of the novel compounds, again, Accelera is probably an exception, but are coming from international initiatives so far. And so somehow figuring out a way to increase the capacity of our Canadian companies to be bold is something which we are thinking about. We also looked at certain items such as clinical trial coordination and management. And it is well known that we have in Canada outstanding clinical trial sites, hospitals, and researchers. But unfortunately, it's also well known that the complexity to start a clinical trial, it is complex to go through the IRBs and to contract clinical trials, and that introduces delays. And it's not just a COVID situation. The situation happens in other diseases, in cancer care and other types of care. And so as we think about pandemic preparedness, can something be done on the clinical trial infrastructure across Canada? Finally, I would also add that we have made recommendations on a series of potential new therapeutics. The landscape is changing dynamically as new trials are being released almost on a daily basis. New trial results are released on a daily basis, which forces CTTF to be quite proactive in tracking things and forces all those stakeholders to be quite proactive. but we probably have to make sure that that proactivity is matched with the ability of the healthcare system itself to actually deliver on certain recommendations so that we have impact on the patients who are suffering from COVID. I think whilst there is a genuine interest on vaccines, and we're all looking forward to a rollout of vaccine on a systematic basis quite significantly, we're all looking forward to more tests. Therapeutics do have a role, as patients unfortunately will, some Canadians will suffer from COVID and some will have severe cases and will benefit from therapeutics. It appears also that the disease is there to stay, the virus is here to stay. So making sure that the new treatments are known to Canadians and they are accessible to Canadians is something which is obviously important to us.
That it? Okay, thank you, Nancy and Cedric, and thank you to all of the speakers, and thank you to everyone participating for the great set of comments and discussions already started in the chat. I'm going to take some of these questions that have been posted and start to shoot them to our different panels and continue to add in your questions. And if you do want to speak, use the raise hand function. For those of you that just got the most recent Zoom upgrade, it's now under reactions, and you'll see the raise hand there. So I'm going to start with the vaccine task force. And we've had a number of comments around, obviously the big issue right now is the limitation of supply. And I realize this may not be directly in the task force mandate, so maybe you could also just explain the relationship with NACI and some of the other groups involved in immunization related work. But the question really is, should we be rethinking our priorities given the short supply that's foreseeable over the next few weeks or months? Either Joanne or Mark?
I can start. So thank you for the question. Yeah, it's hard to figure out which advisory body is doing what thing. So in Canada, we have a federal system, federal government, provincial, 10 provinces and 3 territories. And the provinces and territories are ultimately responsible for delivering programs such as immunization rollout. There is a process federally to provide advice on use of vaccines, and that National Immunization Technical Advisory Group, or NITAG, is called NACI, or the National Advisory Committee on Immunization. It's an independent expert advisory group to the Public Health Agency of Canada. It's existed since the 1970s, and it has a very systematic, evidence-based method for reviewing data. In the case of the pandemic, the situation was there were no vaccines that were approved at the time when the government was trying to procure vaccines. So where NACI reviews authorized vaccines, or vaccines releases its recommendations at the time when the authorization is released, they hadn't in the past looked at future vaccines and things like project management, technical merit of a company, those kinds of things. So that was the unique role of the vaccine task force in relation to other advisory groups, trying to identify, based on principles other than evidence of efficacy in humans or safety in humans, how do you predict what vaccines might be the most likely to succeed? And with regard to the second part of the question, should we be reevaluating? The task force has ongoing meetings where it's looking at its portfolio, determining whether it should readjust, and that is always an option. We're driven by evidence, but there are 4 of the leading candidates, and the other 2 that are in the other 3 that are in the roster for Canada are in phase 3 trials, or have already received authorization. So it is overall a balanced portfolio, and I'll let Mark add anything in there.
Yeah, just maybe a comment on the supply side. And as much, I mean, as co-chair of the vaccine task force, but also as the president of Sanofi Pasteur for 17 years, There seems to be, there is a hypersensitivity, and it's totally understandable, and even a hyper criticism of everything that goes on with COVID. We see that, you all see it, everybody sees it day to day. I almost feel like it's sometimes, if I was still actively employed, it's like my workday would be on the national news every day. And so when we see some of these supply issues, and I know they're impactful, they could have serious, devastating consequences. When something slips from 1 week to another, first week to the third week, well, we'd all like there to be, well, we'd all like that not to happen. That is the way of the world in supplying any sort of vaccines. And you know, what we don't get this week, we'll get the next week, and it does mean adjustments. But I also, my sort of thinking is stay calm and carry on. And one needs to make adjustments as we go, but we don't need to radically change the strategy just sort of day by day. So these things will sort them out. Vaccine manufacturing is very difficult at the best of times, let alone with new vaccines. And so I think we just have to constantly update our information, refine it as we go, not overreact, not underreact, and we will get to where we need to be. It's actually, if you look at it, it's a miracle that we have these vaccines the way we do.
Thank you, Mark. Maybe I'll go to some of the hands that are up, and then we'll go back into the chat. Jacques Tremblay.
Yes, yeah, I just want to say that my laboratory has developed a rapid test for COVID-19. It's based on lamp amplification at a constant temperature. And so we don't need a PCR machine. And the readout is simply the apparition of a fluorescent signal in the little test tube. So I think that's a technology that could be developed and would be very useful for remote area. And also the test can be done in only 1 hour, and it's extremely sensitive. So therefore, I'm looking for potential collaborators to, you know, how do I validate this test and how do I spread its use? And recently there was an article published by Dudna, who received the Nobel Prize for the CRISPR-Cas9 technology. She has developed a technology which is similar to ours, except that she used a Cas13a, which we are also using, but she used a Cas13a, which is more sensitive, and so she doesn't need to amplify to detect the virus. But you know, we are now trying to produce the Cas13a protein that she has produced, and this would avoid one step in our test. We would not have to amplify, but the whole thing is still something which is very simple, done at room temperature, and the apparition of a fluorescent test. So I'm looking for the right person to contact so that I can have some help to validate and spread that test. I think it will be useful.
Well, thank you very much. And I see you posted your email address in the chat, and I'm sure some folks can follow up. And there certainly is the testing secretariat at the federal government. The link was in the, again, in the posting. But maybe, I don't know if either Nancy or Cedric, I know it's not a therapeutic thing, but if either of you have thoughts around, because we have a lot of innovators and entrepreneurs in Canada that have new technologies, and what are some of the things that are being done to help them get them to market?
Yeah, in my case, I'm not an entrepreneur. I'm a university.
You're an innovator.
Yeah, maybe I'll start. And Cedric, you know, we can't speak to the testing side, but for certain groups like IRAP and the Strategic Investment Fund are still open for proposals. That I'm speaking specifically on the therapeutic side, but I think it also exists on the diagnostic side. So I'd encourage you to go to those websites under ISED and have a look at them. Applications are still open for many of those programs. Cedric, I don't know whether you have anything.
Yeah, somebody can send me those links. I will be happy to.
We will send them around.
We'll send them around.
Okay, thank you.
I would also add, beyond the mechanics of it, I mean, a couple of things. First of all, we all know that the business or the work required to bring something to market involves, you know, the classical steps, right? What exactly is the preclinical model? What is the rationale to believe that this would work? So I think spending time, if you have something interesting, to try to access, some samples and do the preclinical models if you can, or at least propose that you will do them if
We
you
had
don't.
access to the viral sequence, and we are able to detect the viral RNA with our test. It's just it's not coming from actual sample. What I would like to do at this point is to compare the results that we are obtaining with our test with the results which are obtained by PCR. We want to make sure that we detect all the positive case and that we don't have false positive. That's what we need to validate this test. And then it could be used rapidly. I think it's simple test. It need very little technical training to be used. Very useful for remote areas.
I understand. I'm not going to talk about the test specifically, but I see what you're saying. I would add, so that's one, the preclinical validation. The other thing I would say is it is quite an intense undertaking to take something from the lab all the way to something approved and clinically used. And so I would almost say, are there people in your network that can help you out, that you can eventually associate with, so that you actually have greater skills around the table? And we have fantastic science, but everything else being equal, the other skills are important, so that to whoever is looking at a potential proposal, either IRAP or SIF or other sources, there's at least the perception that the execution risk is manageable. That's all I would say.
Okay. I'm going to keep us moving along because we have a lot of questions. So Byram Bridle, sorry.
Yes, hello. Thank you for taking my questions. Thank you to everybody for your presentations. I really appreciate this opportunity to be able to ask some frank questions and get some information. So my question relates to, I guess, it would overlap between those who are dealing with the testing as well as the vaccines. So we recognize that we have a shortage of vaccine doses. And so one of the things I'm wondering, and we also know that the naturally acquired immunity following infection with the SARS coronavirus 2 seems to be providing very quite protective immunity and long-lasting immunity. So with that understanding, I have a couple questions. One is, why aren't we doing antibody testing prior to vaccinating, so that we make sure that we are focusing our limited doses on those who have no immunity, rather than risking vaccinating people who already have protective immunity? That would seem to be a waste of doses. Secondly, why I've seen a scenario that causes me specific concern, where I've seen a lot of people now are being tested, and if they come up positive, especially if they fall into a high-risk category, they are being vaccinated about 10 days after their positive test result. That would be the worst time to vaccinate an individual, because when somebody's actively mounting an antibody response, and that would be around, the antibody response would be near its peak 10 days later. We have a negative feedback regulation system built into our immune system that shuts down our ability to respond to vaccines at that time. So that also would seem to be wasted doses. So for those two scenarios, could people maybe provide some feedback, and maybe we need to reconsider vaccinating in those cases, so that we could use our limited doses much more strategically, and therefore accelerate our progress towards herd immunity?
Okay. Joanne, do you want to try?
Sure, I can take a stab at those. So the first question was, why aren't we testing for an immune response before we give people vaccine? And, when you're rolling out a vaccine program, even in a non-pandemic setting, you have to weigh the risks and benefits of everything you're doing to make it as efficient as possible. So from a scientific point of view, we know that people were enrolled in the trials who were already immune and who weren't. So it's safe, and the vaccine still works. So from a safety and efficacy point of view, there wouldn't be any contraindication to do that. From the point of view of rolling out a program, it would be very laborious, and I'm not sure even possible, to procure blood, have it sent off for really neutralizing antibody titers, link that result back to the person who came in for the visit, and then at a separate visit get them vaccinated. That would be probably more effort, I think, than any public health program could manage at this time. And since it's there's no contraindication to doing it. I expect that's probably some of the considerations. And then the second question was about people getting vaccine 10 days after infection. I'm not aware of any recommendation to that effect. That may be an individual practice, but I'm not aware of any national recommendation to proceed in that way. So
Okay, thanks.
that's all I would have to offer at this time.
Okay,
And
thank
there
you.
is
If I could just expand then to the testing side. I do think there needs to be consideration because we could be focusing our vaccination efforts much more effectively. But then on the testing side, comes down to the testing side. So we don't seem to have bothered much in Canada or given much consideration to how
Yeah, so Barb, I showed some data earlier from the seroprevalence studies from the blood banks, which show even late into November that there doesn't appear to be anywhere in Canada where you have natural immunity at a province level above 10%, and natural immunity being infection-acquired immunity. So that's a long way from herd immunity, no matter what you think the threshold is. There may be issues that there's some underestimate related to the nature of blood donors and things like that. And those are probably true, but even when you correct those seroprevalence figures, you're still a very significant distance from herd immunity. So I think it's safe to say that we're not going to get to that threshold by natural infection-acquired immunity alone. But I do think that as we move forward, and some of the work we've been doing on testing is allowing us to understand and detect through testing the difference between infection-acquired and vaccine-acquired immunity. And that may be helpful if we can roll that out in surveillance to understand how far we are towards herd immunity. So it's definitely an important part of the big picture.
Yeah, thank you very much. Just one point of clarification, though. What's the youngest that somebody can? What's the youngest age at which somebody can donate blood? Because I rather suspect, with all of our children having been back at school, that's probably a very large population where we could be particularly enriched for natural immunity.
You're right. The age for blood donors is 17.
Yes.
So we don't get that in those studies, but we do have some other studies underway which are trying to shed some light on younger kids.
So I would argue we may have dramatically underestimated, especially in that demographic.
Yeah, Tim, could you just say a few words, because there was a question in the chat about random sampling, and I don't think you said much about the StatsCan work.
Yes, so I was reporting on convenience sampling, but the household-based sampling, which is random and more representative of the full population, is something that we rolled out with StatCan, and the surveys in the field. It's a household survey that's in round one, and many citizens are receiving the test through the mail as we speak. And this will give us, I think, a better picture across all age groups and across the entire country of what are called a more scientific approach to sampling of the population. So that's, I think, one thing that we have to look forward to very soon. And we've also geared the StatCan study to be sensitive to whether people have been vaccinated or not. So I think we're going to we'll be able to tease that out as well. The one thing I would say is, and this is where there's probably lots of epidemiologists on the call, but the distribution of infection is very lumpy. And so I think this is why targeting in on high-risk areas or populations is a critical complement to understanding the bigger picture. And sometimes that's that's a different sampling strategy, but just to say the CITF has been supporting studies on that front as well.
And Tim, there was also a query about availability of the data. And is it safe to say that there will be a process for ensuring that modelers and others will be able to get at the data?
Yes, so David Buckridge is the person who's in charge of our data sharing agreements with all of the studies, and he's also part of the PHAC modeling group. And so he's working with the modelers as much as possible to get access to the data that is going to enhance the precision or feed the models. So we're trying to accelerate that connection as much as possible. But David Buckridge, who works with me at McGill, is part of the modeling group, is the entry point for that.
Great, thanks, Tim. So I'll go to Michael Wolfson next, and I'll ask people to try to keep their questions brief so we can get as many in as possible.
Sorry, just a second, I click here. Yeah, quick question to follow up on the vaccine surveillance, maybe to Tim, and that is, how are the data flows going? For example, are they adequate, and are you running into any problems merging data sets across, for example, jurisdictions?
Michael, I think as somebody who worked at StatCan for as many years as you did, you probably know the answer to the question. But there are all sorts of issues as you know, because Canada doesn't have a unified vaccine surveillance system. We're working with some of the great assets that have been developed over time to provide a pan-Canadian picture, for, most specifically in the context of CERN, the Canadian Immunization Research Network. But there is also an effort that the federal government, PHAC, has mounted on getting an IT system that will align different dimensions of the vaccine rollout, from logistics to coverage. And we've had discussions with them, and their system seems to be well developed. And so I think some of the architecture for bringing the data together is falling into place. But this vaccine surveillance reference group, one of the reasons that it's been put together is to try and align those studies a little bit more at the outset, so that we will have efficient ways of pooling and keeping an eye on the big picture of the various issues related to safety and efficacy or effectiveness that are going to be monitored in those studies. But the simple answer is it's complex because there are going to be many different actors, and we'll try and do our best in terms of pulling it together.
Thanks, Tim.
I remain concerned that there's kind of this privacy chill that's blocking things, and my understanding of the IT stuff that PHAC is buying, or Health Canada, is that it's only for the federal jurisdiction, so it's not clear that the provinces would pick it up.
Just to say, Michael, I think those are valid concerns, although in my discussions with the folks at PHAC that are leading on that, I had the impression that they're having very productive discussions with the provinces. So I don't know what that's going to lead to, but they seem to be optimistic.
Okay, I'd like to just ask Irfan, has been very busy answering questions in the chat. Is there anything that's been in the chat that you want to comment on further?
I think there's this interesting debate about whether rapid antigen tests are actually the gold standard when it comes to trying to determine whether somebody is infectious. So, there's a view that the PCR, or not a view, I mean, the PCR tests do stay positive long after somebody is no longer infectious. And while the rapid antigen tests have a higher false negative rate than the PCR, there is a school of thought that those false negatives aren't really false negatives because the people with the so-called false negative wouldn't transmit the virus. So I'll say, I think that's an interesting debate. I'm not an infectious disease physician or a medical microbiologist or a virologist. I'm a general internist. I feel like, you know, Supesh and my role on the expert panel is mostly to steer the discussion and make sure that we're, you know, we have enough breadth and depth on the panel and are consulting widely. So I don't know the answer to that question. I'm not sure that anybody truly does. It's clearly an issue of ongoing debate. So there's some interesting comments on that in the chat, including some anecdotal data from an infectious disease physician I spoke with earlier this morning.
Thanks,
That's probably the only thing I'll highlight.
Yeah, I think you said rapid antigen tests are the gold standard. I think the issue is that a lot of these studies, including what you've just posted, are comparing rapid antigen tests and PCR, and neither one of them is actually the gold standard of whether someone is infectious. And you know, there's only we really do not have a proper gold standard. And again, it's not surprising. We're only a year into this disease, and so we still have a lot left to learn. Okay,
that's a better way of putting it. Thanks, Vivek.
I'll go to Bonnie Mallard.
Yeah, hi there. Bonnie Millard. I'm professor of immunogenetics at the University of Guelph. And I'd just like to comment a little bit more on the percent seropositive discussion that's been going around. And it does seem quite low because if you just sort of looked at the data, even from today on COVID mapping, we've got about 688 688K people who have recovered so far. And if you just even divided that over the 38 million for our population, that would give us about the 1 to 2% seropositives that I think, if I didn't miss out, that was kind of the realm of the seropositives. So it seemed a bit low. And then I saw that, I think it's Stephen Pellich had commented that his test, and I'm not familiar with that, from BC, showed higher seropositive. I'm not sure if he's on the call and could tell us what he means by a lot higher. I'd be curious about why we're seeing those differences.
So maybe I'll invite
Yeah.
Steve. Yeah,
I'm here. So basically, we developed a test from last February where we took SARS-CoV-2 virus genome sequence and made about 5,000 peptides, made spot arrays, tested serum now from, I guess it's pretty close to 50 covered COVID-19 patients, and as well as about 100 healthy controls. We found that the results were that about 120 peptides were pretty good for picking up different regions. These peptides are based on short sequences within the protein sequences of those, pretty much all the proteins that were in the virus. And we found that actually the immune response was completely different for every COVID-19 patient that we looked at. And that when we took pre-COVID samples from 2019 and early 2019 and earlier, we were finding positive results with our tests in those samples. Plus, in people who are associated with people who've had COVID-19 and tested positive, we found that they had antibodies. And again, the responses were very variable. We also tested babies, serum from babies, and we get no detection of antibody responses. So all the data that we have points to actually either SARS-CoV-2 who has been around a lot earlier in BC, which I suspect is true because we didn't have any restrictions for about 3.5 months when the virus probably first showed up in Vancouver in early December. Restrictions only really happened in mid-March. So I suspect either it's spread around or there is other SARS-CoV-2-like viruses that have been in the population that has given some immunity, and that might account for why this virus is asymptomatic in most people that are certainly not in a high risk group. we've only had about 12 deaths in BC for people under age 50 this entire time. And that accounts for about 70% of the positive tests from the genetic tests. So I see a big discrepancy here. And
So what was
that's
the
why
percent seropositive overall, if you could give a number to kind of compare to what had been presented?
I would say that of the so-called healthy people that we've tested, which includes myself, my staff, a lot of other people that have volunteered in a clinical study, what we find is that it was probably close to at least 50%. We have evidence of multiple antibodies against different epitopes in the same person.
Okay, thank you for that. I look forward to hearing more as you continue that study. One other quick
Yeah.
question that I've been getting from my students, and I just love to be able to give them an answer. There was a lot of talk about changing the vaccine protocol because of the shortage of vaccines. And sometimes I hear, yes, we will consider changing that and making it the booster at a duration that is not recommended by the manufacturer. In other words, greater than 21 days with Pfizer, 28 days with Moderna. What is the current status on that? Are we considering changing that, or are we not?
So I'll turn it over to Tim. You might address the first, and you want to take a stab at the second one as well?
Yeah, Bonnie, thanks for your question. I think one thing that's important is that the seroprevalence that came out from Canadian Blood Services was not for the month of January. It was for November. So you need to make sure you compare the expected rate based on the numbers infected in November. However, even when you do that, it suggests that there isn't a big difference, in large part because of the results in Ontario, Ontario being the biggest province. And there's been no measured change in seroprevalence using the convenient sample of blood donors in that province. And that requires a lot of further investigation because that doesn't correspond with the evolution of the epidemic. So one really needs to look at things. However, in the Prairies, in October and November, if you do the math based on seroprevalence at 8% and 8 or 9% in Manitoba, for example, and compare to the numbers infected then, you see about a 10-fold difference with the measure of seroprevalence. And this is what you see in a lot of international studies. So there's more infection going on that's being detected by PCR, the nasal swabs, but even when you detect that 10-fold increase, it still it doesn't get you above 10%, at least in November, in provinces that had widespread community transmission. So that's still a long way from herd immunity.
Maybe I can see Kate's face here. She wrote an op-ed on the dose interval piece, if she wants to comment on it. Kate is one of the co-chairs of the Immunity Task Force.
Yeah, so we are, thank you, Vivek. We're following generally in Canada the NACI recommendations, which are also inspired by the WHO recommendations that say up to 42 days from the 21 with Pfizer or the 28 with Moderna. There's a lot of debate in the vaccine community about whether that's ideal or not, and whether that kind of delay actually gives you a better response or not. It's a bit of a natural experiment, and hopefully we'll be able to track those differences. We see things like Quebec going for 90 days. We see each province seems to be deciding what they're going to do. So hopefully with the Vaccine Surveillance Reference Group, we'll be able to track this and actually see what differences it might make. And hopefully we can get our vaccine supplies flowing very rapidly so that we don't have to do that.
Yeah, thank you for that, Catherine. I particularly because as an immunologist, hate to see Canadians sort of as part of an experiment. And as an experienced person working with vaccines, I also know we have no guarantee how they work if we don't have any data when we change the protocol. I think this is going to send a lot of alarm bells throughout Canada.
Okay, we just have a few minutes left, and I see Byram, you have your hand up again. You have a quick follow-up comment?
I do, yes. I just want to reemphasize my concern with this consideration of altering protocols as well. Without data, it's potentially very dangerous. And I want to say I've spent years and years developing all kinds of vaccines. And one of the things that a lot of people don't realize, and unless you do this work, is, again, we don't know exactly how these vaccines work, but certainly the first vaccine would be designed to induce a primary response. The second one, in this close interval, is kind of like this suboptimal effector response that often correlates with a suboptimal memory response as well. And in fact, by suboptimal, that also means it could have a tendency to wane quite quickly. So one of the things we have to keep in mind is that with that 3 to 4 week interval that's been recommended, that might be targeting this weak sort of suboptimal memory response. And if we delay these intervals, we're going to miss that memory response altogether. And therefore, that second dose will be the equivalent of having given somebody a single dose. So again, without the data and without it being done in a phase 3 trial and tested, and it's not just me. I mean, the FDA, US FDA, which I think we have to give a lot of respect to, is insisting that this should not be done. So as an immunologist with lots of experience with this, I would tell all Canadians that they have to insist on the vaccine being administered as it was approved. Otherwise, there's no guarantee of efficacy or safety.
And maybe I'll just add that, those kind of concerns are among the risks and benefits of changing the interval. The Chief Medical Officers of Health and NACI both have said that in the first instance, one should aim to keep the second dose within the range that it was approved, but in extraordinary epidemiological circumstances, one could go out to this longer time point of 42 days. But I think the emphasis is keeping to the protocol as it was done.
Yeah. So we just have a couple of minutes left, and there were a number of questions or comments about surveillance for the variants. And I think it's just, these are the 4 task forces that are up and running. Variants is a relatively new issue, and so there isn't yet a task force on the variants. But first of all, does anyone want to comment on it, or I'm happy to make a few comments as well. Nancy?
Maybe I just offer that there are discussions with all the co-chairs of this group as well as other groups, and Vivek might open this, that's been ongoing since the topic of variants has been raised. And obviously the majority of people on this call would acknowledge that we all knew that variants would be coming. And so as part of our follow-up from the CTTF, we actually have been requesting additional data and following that data as it relates to therapeutics and the variants. So we've been proactively asking that question around the programs that we've looked at and advised on and continue to look at. So we're aware and obviously keeping informed is what I would say.
Cedric?
I would also add, that's why it is important, a couple of things. It's important to have and to keep on working on the portfolio of therapeutics. Therapeutics, of course, could lose, depending on mode of action, efficacy on variants, but some therapeutics are variant, or so far as we know, variant agnostic. For instance, the dexamethasone, which is widely used in the severe cases. And so I think as a country, it is quite important that we keep on pushing on, obviously, improved testing and tracing, number one. Number two, genetic sequencing done on a more systematic basis so that we do detect variants, because they may actually not only be imported, but emerge from Canada itself. And then, of course, evolving our diagnostics and therapeutics as variants emerge.
Thank you. Unfortunately, we're at time, and I'll just note that there will be and Fatima just posted a link to the Genome Canada work for CanCOVID. There's a number of groups, as was noted, that are working on this, and you will be seeing more from CIHR and other agencies on the approach that's going to be taken. So I want to just close by thanking all of our presenters for preparing their presentations and keeping to time. The wonderful discussion that we've had and the chat. We will be saving the chat and going through and if we can follow up on some of these questions, we will follow up. I'll remind everyone, because there's many comments about connecting with people in other research areas. That's why CanCOVID exists, cancovid.ca. And if you go to the website, there's a set of tools there, including our Slack platform that can help facilitate finding researchers and collaborators in other areas. There were a number of questions and comments about potential research funding. And again, I can't promise it, but my understanding is that there is work being done on potentially having another round of CIHR special call for proposals for targeted research. And then finally, we will work with the presenters and get a recording and the slides posted on the CanCOVID site, and Rosa's posted the link there for you. We have a number of other workshops like this that we're starting to plan, and look forward to seeing you at future sessions. Thank you very much.